variolation, an early Asian method which introduced dried scabs of smallpox patients and was later modified in Europe. So ano yung variolation? Ang ginagawa nila before is kinukuha nila yung dried scabs. Ano yung dried scabs? Halimbawa, nagkaroon kayo ng sugat or nagkaroon kayo ng chickenpox, nagkakaroon siya nung, pag natutuyo na siya, nagkakaroon siya nung parang black na part na indication na natutuyo na siya or siya yung parang dried skin. So, kinukuha nila yon and then, it was introduced to patients. Okay? So, parang siya yung nagiging pantapal doon sa mga taong may smallpox. So, parang kinukuha nila mula doon sa mga taong gumaling na and then nilalagay nila doon sa mga taong meron pa. Okay? Ano yung smallpox? Yung smallpox ay isang sakit na cause ng isang viral infection. So, para siyang, kung may imagine nyo, ang itsura niya para siyang chickenpox, kung nagkaroon na kayo ng chickenpox. Or yung bulutong. But this one, ang blisters niya medyo mas malalaki. So, hindi siya small. Big ang blisters niya. Okay? During that time, maraming taong nagkaroon ng smallpox. So, it became an epidemic. This method ng variolation ay ginamit nila para malaman kung effective ba siya. Nakukuha ka nung scabs or yung natutuyong balat mula doon sa mga blisters and then ilalagay mo siya doon sa infection naman ng mga taong parang early pa yung kanyang stage ng blisters niya. This was first tested among abandoned children and prisoners. During the earlier days, yung mga experiment nila sa tao, ginagawa nila mostly sa mga prisoners. When it was declared safe, the members of the English royal family were inoculated with this variolation. But then, it was a scientist named Edward Jenner who really was the first person who made a vaccine. So, during the time, variolation yung ginagawa nila. Lalagyan mo siya ng scab and then ipapatong mo siya doon sa merong mas maagang blisters or mas early symptoms ng blisters niya. Pero this time, si Edward Jenner, naka-invent siya ng way to fight smallpox. Yung smallpox is a disease that is very deadly. So most of the patients na nagkakaroon nito, actually a big percentage ay namamatay. So during the time ni Edward Jenner, meron mga milkmaids. Okay, ano yung milkmaids? Sila yung mga kumukuha ng milk from cows. Okay? Napansin ni Edward Jenner na yung patients niya na mga milkmaids ay nagkakaroon ng isang mas mild version ng smallpox. Ito yung tinatawag naman na cowpox. Okay? Ulitin ko. Yung mga milkmaid during the time, napansin ni Edward Jenner na nagkakaroon sila ng milder disease which is cowpox. And then, pag nagkaroon sila ng cowpox, hindi sila nahahawa ng smallpox. Yung smallpox siya yung mas deadly na disease. Okay? And then what happened is parang sinabi niya na bakit kaya yung mga milkmaids na yon, dahil nagkaroon na sila ng cowpox, hindi na sila nagkaroon ng smallpox disease. So, inisip niya na possible na kapag nagkaroon na ng cowpox disease, ay nagde-develop na siya ng mas malakas na antibody na po pwede ring lumaban against smallpox. So, in his experiment, he injected a farm boy with a cowpox. So, this boy is actually a relative of Edward Jenner. So, pinag-eksperimentohan niya yung sarili niyang apo during the time. And then, nilagyan niya ng cowpox. Okay? Yung virus ng cowpox. In-inject niya dun sa apo niya. And then, after some time, dahil nilagyan niya ng virus, yung bata, nag-develop siya ng cowpox disease. After that, gumaling yung bata from the cowpox. So, ang ginawa niya, in-inject niya naman ng isa pang virus, which is from the smallpox, dun sa bata. Okay, so this is actually an experiment dahil hindi niya alam baka mamaya mamatay yung bata because of the virus. So yung experiment niya na yun, hindi niya rin talaga alam kung ano yung kaya niyang i-expect. So ang in-expect niya lang is dahil nagkaroon siya ng cowpox, magde-develop din siya ng resistance against the smallpox virus. So in-inject niya yung smallpox virus dun sa bata. Anong nangyari? Okay, the boy resisted the disease. Ang nangyari is nag-develop yung bata ng resistance ng antibody against the deadly smallpox disease. Because of their similarities, vaccination with the cowpox virus synthesizes the immune system to react vigorously if exposed to actual smallpox virus. Effective vaccines against many other viruses exist after the experiment of Edward Jenner. So ulitin ko, ano nangyari doon sa experiment ni Edward Jenner? During the time ni Edward Jenner, napansin niya maraming milkmaids ang nagkakaroon ng cowpox. Pero hindi sila nahahawa ng smallpox virus na mas delikado at nakamamatay. So ang ginawa niya, ineksperimentohan niya yung isang bata, nilagyan niya ng cowpox virus, and then yung bata nagkasakit because of the cowpox. Okay, pero nag-recover siya kasi mas mild yung sakit na cowpox. After some time, nung gumaling yung bata, nilagyan niya naman nung deadly na smallpox virus. Fortunately, the boy developed resistance against the virus. So dahil doon, nalaman niya na a milder virus na nagdudulot ng cowpox can induce resistance doon sa mas malakas na virus ng smallpox. And this gave rise to what we call as a vaccination. I would like you to watch a short video regarding these things that I am saying about the first vaccine na dinevelop and then pinarami and then in-inject sa mga tao during the time of Edward Jenner.
Ten 10,000 years ago, a deadly virus arose in northeastern Africa. The virus spread through the air, attacking the skin cells, bone marrow, spleen, and lymph nodes of its victims. The unlucky infected developed fevers, vomiting, and rashes. 30% of infected people died during the second week of infection. Survivors bore scars and scabs for the rest of their lives. Smallpox had arrived. In 1350 BC, the first smallpox epidemics hit during the Egypt-Hittite War. Egyptian prisoners spread smallpox to the Hittites, which killed their king and devastated his civilization. Insidiously, smallpox made its way around the world via Egyptian merchants, then through the Arab world with the Crusades, and all the way to the Americas with the Spanish and Portuguese conquests. Since then, it has killed billions of people, with an estimated 300 to 500 million people killed in the 20th century alone. But smallpox is not unbeatable. In fact, the fall of smallpox started long before modern medicine. It began all the way back in 1022 AD. According to a small book called The Correct Treatment of Smallpox, a Buddhist nun living in a famous mountain named Ome Shan in the southern province of Sichuan would grind up smallpox scabs and blow the powder into nostrils of healthy people. She did this after noticing that those who managed to survive smallpox never got it again, and her odd treatment worked. The procedure, called variolation, slowly evolved, and by the 1700s, doctors were taking material from sores and putting them into healthy people through 4 or 5 scratches on the arm. This worked pretty well, as inoculated people would not get reinfected, but it wasn't foolproof. Up to 3% of people would still die after being exposed to the pus. It wasn't until English physician Edward Jenner noticed something interesting about dairymaids that we got our modern solution. At age 13, while Jenner was apprenticed to a country surgeon and apothecary in Sodbury, near Bristol, he heard a dairymaid say, "I shall never have smallpox, for I have had cowpox. I shall never have an ugly pockmarked face." Cowpox is a skin disease that resembles smallpox and infects cows. Later on, as a physician, he realized that she was right. Women who got cowpox didn't develop the deadly smallpox. Smallpox and cowpox viruses are from the same family, but when a virus infects an unfamiliar host, in this case cowpox infecting a human, it is less virulent. So Jenner decided to test whether the cowpox virus could be used to protect against smallpox. In May 1796, Jenner found a young dairymaid, Sarah Nelms, who had fresh cowpox lesions on her hand and arm, caught from the udders of a cow named Blossom. Using matter from her pustules, he inoculated James Phipps, the 8-year-old son of his gardener. After a few days of fever and discomfort, the boy seemed to recover. 2 months later, Jenner inoculated the boy again, this time with matter from a fresh smallpox lesion. No disease developed, and Jenner concluded that protection was complete. His plan had worked. Jenner later used the cowpox virus in several other people and challenged them repeatedly with smallpox, proving that they were immune to the disease. With this procedure, Jenner invented the smallpox vaccination. Unlike variolation, which used actual smallpox virus to try to protect people, vaccination used the far less dangerous cowpox virus. The medical establishment, cautious then as now, deliberated at length over his findings before accepting them. But eventually, vaccination was gradually accepted, and variolation became prohibited in England in 1840. After large vaccination campaigns throughout the 19th and 20th centuries, the World Health Organization certified smallpox's eradication in 1979. Jenner is forever remembered as the father of immunology. But let's not forget the dairymaid Sarah Nelms, Blossom the cow, and James Phipps, all heroes in this great adventure of vaccination, who helped eradicate smallpox.
Okay, did you understand the story of vaccination? Or do you have questions?
Yes.
Hindi yan.
Miss, we understood po.
Okay, so because of this, wait lang balita ko yung pinapresent ko. So because of what Edward Jenner did during his time, smallpox virus was completely eradicated on the face of the earth. So until now, wala na kayong nakikitang mga cases ng smallpox na sakit. So yung usually ang meron na lang is the chickenpox and other diseases. So yung smallpox, dahil dun sa ginawa ni Edward Jenner, unti-unti naparami yung vaccine na yan. And then, eventually, it was eradicated on the face of the earth. So, tinrace nila yung pinakahuling cases ng smallpox. So, ngayon, wala nang hawaan na nangyayari or wala nang cases ng smallpox sa buong mundo. But there are actually 2 strains or meron pang dalawang strains ng virus na yan na nakatabi. Okay, one is in America, one is in Russia. So some scientists, kinakabahan sila baka mamaya during wars or in some other time in the future, magamit yung virus na yan as a biological weapon against some people. Katulad ng nangyayari sa nangyayari ngayon na pagkalat ng COVID-19. So, natatakot sila baka mamaya yung mga nakatabing strains pa ng virus na yan ay maikalat uli and then mag-cause uli ng death since siya ay delikado and very deadly. Okay? But for now, nakatabi yung virus na yon and wala na tayong cases ng smallpox around the world. And this started vaccination. So this was Edward Jenner in 1796 when he discovered that cowpox or vaccinia come from the word vaca or cow. Okay? Dahil nga ang pinagsimulan niyan is yung cowpox or yung galing sa na-observe nila na sakit ng mga cow. So vaccinia comes from the word vaca or cow. So this induced protection against human smallpox and then tinawag niya itong vaccination. And then because of this, Edward Jenner is termed as the father of immunology. Vaccination is the inoculation of healthy individuals with weakened or attenuated forms. Pag sinabi nating attenuated, ang ibig sabihin nun weakened, mas mahina na form ng microorganism that would otherwise cause disease to provide protection or active immunity from disease upon later exposure. So, yung vaccination, ito yung mas mahinang virus katulad nung ginawa ni Jenner, na cowpox muna yung nilagay niya dun sa bata and dun sa ibang tao. Okay? Yung virus na yun, dahil magkapares yung itsura niya, in-induce niya yung antibodies or yung resistance ng bata against the type ng virus. And then pag nakapasok na yung mas malakas na type ng virus, which is the smallpox virus, meron na siyang antibody against that virus and then it provided immunity. Immune na siya doon sa sakit na yon and then hindi na siya nagkakaroon ng signs and symptoms nung sakit na yun dahil nga meron na siyang antibody and then dahil nga immune na siya yung katawan niya against doon sa type ng virus na yon. So, vaccination is weakened or attenuated form ng virus or ng microorganism. So this one is the history of vaccines. So this one is a timeline kung kailan unti-unti na-discover yung mga different vaccines against different diseases. So in 1813, the Congress creates a National Vaccine Agency. And then in 1855, the MA passes the first law of US mandating vaccination for school kids. So ngayon, 'di ba, baby pa lang bina-vaccinate na against tuberculosis. Meron yun sa mga school children, bina-vaccinate sila against hepatitis. Kasi nga may mga laws na nagsasabi na kailangan i-vaccinate sila to protect them against viruses na po pwede nilang makuha sa school and their environment. In 1886, the typhoid vaccine was produced. 1885 is the rabies vaccine. 1891 is the glycerin used to reduce germs in vaccines. In 1896, the cholera vaccine. Okay, kung matatandaan nyo, may diniscuss na ako earlier kung sino yung naka-discover niyan. In 1926, the discovery of aluminum salt as adjuvant sa vaccines, pertussis and TB vaccines, diphtheria vaccine, tetanus vaccine. O kapag nagkakaroon ng sugat, usually pinapa-inject ng tetanus vaccine that is to protect them against this bacteria. Every year, usually nagkakaroon ng influenza vaccine. Yellow fever vaccine, polio vaccine. In 1955, there are a lot of children who became paralyzed dahil sa polio. And another scientist ang nakadiscover ng vaccine na yan against polio, kaya nabawasan yung infantile paralysis or yung pagpaka-paralyze ng mga maliliit na bata due to polio. Mumps vaccine, rubella vaccine, and then anthrax vaccine. Measles vaccine, ito kung matatandaan nyo, yung mga babies, binibigyan sila nito, usually mga 9 months, 1 year, binibigyan na sila ng measles vaccine or yung tigdas. last year actually nagkaroon ng scare against against vaccines. So yung mga nanay, marami sa mga nanay hindi na nila pinalagyan ng measles vaccine o yung mga anak nila. Bakit nagkaroon ng scare against this vaccine is because of the Dengvaxia. Sinasabi nila na yung mga na-vaccinate ng Dengvaxia ay namatay. Okay, yung mga school kids na na-vaccinate ng Dengvaxia, namatay sila because of the vaccine. But actually, hindi naman pa talaga napapatunayan na it's because of the vaccine kaya namatay yung mga bata. And dahil doon nagkaroon ng scare, yung mga nanay napabakunahan yung mga anak nila ng measles vaccine. Kasi baka nga mamaya mangyari din yung nangyari doon sa mga batang namatay na nabakunahan ng Dengvaxia. So, ang nangyari, marami ngayon yung mga bata last year na namatay din dahil sa measles. Okay? So, delikado rin yung mga ganyang sakit, lalo sa mga bata na mahina pa ang immune system nila. So, kailangan binavaccinate sila to trigger yung pag-produce ng mga antibodies against these viruses. And then in 1974, meningitis vaccine, 1977, the pneumonia vaccine, 1981, hepatitis A, hepatitis B vaccines. In 1995, the varicella zoster vaccine, yun yung para sa chickenpox. Okay, earlier, actually during our days na mga bata pa kami, hindi pa uso yung mga chickenpox vaccine. So, ang nangyayari kapag nagkaroon ng bulutong or chickenpox, napakarami talagang blisters na lumalabas. So, sobrang sakit kapag nagkakaroon ng chickenpox. You can ask some of your mga older siblings o yung mga nanay nyo kung ano yung itsura before ng mga taong nagkakaroon ng chickenpox before nagkaroon ng vaccination. So, in 1995, nagkaroon na ng vaccine against chickenpox. So, bihira na natin makita yung mga bata na napakaraming blisters dahil dun sa chickenpox. Kasi nga, nare-reduce nito yung symptoms ng chickenpox because vaccinated kayo. In 1999, the rotavirus vaccine was made. Ito yung para naman sa mga babies na to protect them against viruses and bacteria that may cause harm dun sa kanilang digestive tract and cause severe diarrhea. In 2006, HPV or human papilloma virus and shingles vaccines were introduced. So, if you can look closely on this timeline, unti-unti nagiging mas malapit na yung pagka-discover ng mga vaccines against different microorganisms. So, makikita nyo, in this 1950 hanggang 2000, ang dami ng mga na-discover ng mga vaccines against different diseases. And it all started with the smallpox vaccine that is made by Edward Jenner. Later on, I will go back to this timeline because this will be in coordination with your performance task. After Edward Jenner, this is Pierre Paul Emile Roux. Together with Louis Pasteur, he reported that incubating cultures longer than normal in the laboratory resulted in attenuated bacteria that could no longer cause disease. So after ng work ni Jenner, na-discover naman nila na even ang mga bacteria, pwede natin gawa ng vaccines. So, sabi niya, yung attenuated, pag sinabing attenuated, weakened, mas mahinang bacteria, could no longer cause disease. So, working with chicken cholera caused by Pasteurella multocida, they noticed that animals injected with attenuated cultures were resistant to the disease. So, siya naman, na-discover niya yung attenuated bacteria. Okay, so yung kay Jenner, these are attenuated viruses. Si Roux naman and Pasteur, na-discover nila yung attenuated bacteria, mas mahinang form ng bacteria. And then, tinest niya yon doon sa chicken cholera that time. And then, na-discover nila that it produced immunity or resistance doon sa sakit. So, this is Pierre Paul Emile Roux or Roux na tumulong sa paggawa din ng vaccines against naman sa chicken cholera. These are Emil von Behring and Shiba Saburo Kitasato. Okay, this one is in 1845 to 1831. They said that antibodies raised to inactivate diphtheria toxin by injection different host, a rabbit with a toxin of a toxoid form, and then anti-serum recovered contains antibodies specific for the toxin and protection from disease when injected with non-immune subjects. So ito yung kanilang discovery naman is about the diphtheria. Okay? Yung vaccine naman against diphtheria. Okay? Ito kailan to binibigay? Usually binibigay ito sa mga bagong panganak or kapag kailangan. Usually yung mga baby na bagong panganak, binibigyan sila ng DPT vaccine, that is diphtheria vaccine, na panglaban against this diphtheria microorganism and then dun sa toxin na pinoproduce ng bacteria na yon. Okay, so si Behring, Emil von Behring, and Shibasaburo Kitasato, they work on the vaccine or immunization against diphtheria. This is Charles Chamberlain, and he developed other vaccines together with Louis Pasteur. This one is yung nabanggit ko in the earlier discussion ko na na-discover ni Pasteur yung anthrax and anti-rabies vaccine. So, tumulong doon si Charles Chamberlain. So, ang discovery nila is attenuated anthrax vaccine through chemical and heat treatment. Yung chemical na ginamit nila is potassium bichromate. And then, attenuated rabies vaccine. He propagated the virus in a rabbit following the injection of infected brain and spinal cord extracts. So, ito yung ginawa nilang experiment dun sa kanilang development of the vaccines ng anthrax and rabies. So, yung anthrax before, nagkaroon actually ng scare niyan. Kasi nagkaroon in the history na inispread yung anthrax na yan sa mga politicians. So, hindi rin nila alam kung kanino galing yung bacteria na yon, pero nilagay siya sa mga male. Okay? Hindi sa lalaki, sa mga sulat. Before kasi mauso ang internet, ang emails, usually snail mail ang ginagamit para maghatid ng information. Okay? So, during that time, yung snail mail, yun yung hinuhulog sa post office. And then, hihintay ka ng a few weeks bago mo siya makuha, ibibigay siya ng postman dun sa bahay nyo. Okay? Before, that's the form of communication. So, ang nangyari in history is that yung mga politicians, this is in other country, naka-receive sila ng mga mail, okay, ng snail mail. And then, pagbukas nila ng snail mail, siya pala ay infected ng anthrax. Anthrax bacteria, which is Bacillus anthracis. So, eventually, nagkaroon sila ng sakit because of that. Okay? So, it can be spread on males, on snail mails, and on other surfaces na infected. Okay? But there is a vaccine already for anthrax na dinevelop ni Chamberlain and Louis Pasteur. And then also, there is also already a vaccine against rabies na which is very helpful before napakaraming namamatay because of rabies. Actually, in the early 1990s, maraming namatay against rabies dito sa Philippines. Dahil hindi nila sineryoso yung kagat ng aso na yan. Or actually, the real reason is that mahal ang rabies vaccine. And ilang beses yang i-vaccinate. Usually 2 to 4 times kang babalik. And then nasa 2 plus or 2 or more vaccines ang ilalagay sa'yo per visit. So actually mahal ang rabies vaccine before. Kaya maraming namamatay na dahil sa rabies virus. But now, we are very lucky na medyo mas mababa na yung presyo niyan ngayon. Plus, available na siya usually sa mga hospitals and sa mga clinics. So, if ever nangyari nakagat kayo ng aso na hindi nyo alam kung rabid ba or hindi, it's better na magpa-vaccinate agad. We have vaccines sa mga city hall, sa mga clinics, and then, of course, sa hospitals. Sa City Hall, I believe mas mura na yung babayaran or minsan libre yung vaccines na yan. All because of these people who helped develop the vaccine. This one is Erlich in 1845 to 1916. He discovered bacteria engulfing phagocytotic cells in the blood. So ano yung phagocytosis? Phagocytosis is also known as cell eating. Okay? Ito naman ang na-discover niya ay ang cellular immunity. Ano yung cellular immunity? When phagocytic cells get to the invaders, for example, bacteria, they go through the process of phagocytosis to finally eliminate the bacteria. Anong ginagawa? Anong immune system natin o yung ating cells? Or particularly the white blood cells. Anong ginagawa nila? Ini-ingest nila yung bacteria na yan na nakakapasok sa ating blood. Okay? So, ang ginagawa, ini-ingest niyan and in the process, pinapatay nila yung bacteria. Okay? Malalaman nyo na nagkakaroon ng laban. Okay? It's actually like a war against this bacteria dun sa inyong katawan kapag yung mga lymph nodes nyo ay namamaga. Ano yung lymph nodes sa mga kulane? Meron kayo niyan sa may leeg or sa may singit. Pansinin nyo kapag meron kayong sipon, masakit yan. Okay? Sa may malapit sa tenga. Okay? It is because these cells are fighting this bacteria or viruses and then help eliminate them on our body. So may mga sakit na hindi naman natin kailangan talagang magpa-vaccinate, such as common colds or yung sipon. Common colds is actually caused by a virus, which is also another form ng coronavirus or different types ng virus. Every time na nagkakaroon kayo ng sipon, iba't ibang type ng virus ang nagkakos nun. Okay? Pwedeng coronavirus, pwedeng a different type of virus, and then hindi siya laging pare-parehas. So, possible na simula nung maliit kayo, sipunin kayo, or nagkakaroon kayo lagi ng colds. Pero iba't ibang uri ng virus ang nagkakos nun. Okay? Kasi every time na nagkakaroon kayo ng sipon, the virus is, the immune system fights this virus, and then ini-engulf na yon, pinapatay nila yung virus na yan, para next time, hindi na kayo uli maapektuhan ng virus na yan. Pag nagkaroon kayo uli ng colds virus, possible na ibang type naman siya ng virus. Okay? So, another way in which our body fights these microorganisms is through cellular immunity with the help of these phagocytic cells na kumakain at nag-eliminate ng mga bacteria and some viruses na nakakapasok sa katawan natin. Kaya minsan kahit hindi tayo magpa-vaccinate, gumagaling naman tayo, lalo dun sa mga viruses na nagkukos ng colds or yung common colds. Okay? Because our body is able to fight these microorganisms. Ito yung reason kung bakit sinasabi nila na dapat laging malakas ang immune system mo para hindi ka magkasakit. Okay? Or yung resistance mo. So, ito yung reason nun. Dahil pag malakas ang immune system mo, malakas ang soldiers ng katawan mo, kaya nilang labanan ang mga viruses, ang mga bacteria na nakakapasok sa katawan. Pag mahina ang immune system, wala silang panlaban against this microorganism. So, madali silang nagkakasakit, and some persons mas severe yung effect sa kanila ng sakit na yon. So, kaya nga 'di ba ngayon hindi muna pinalalabas yung mga mas bata at saka yung mas matanda kasi sila yung compromise yung immune system. Kasi possible na sila yung mas magkakasakit kasi medyo mas mahina yung panlaban nila against these microorganisms. Okay, the one who discovered these phagocytic cells is Erlich, and he said that these are evidence ng cellular immunity. And then we go now to John Snow in 1800s. Okay? Si John Snow naman is known as the father of epidemiology. Epidemiology is come from the word epidemic, wherein a disease spreads from one person to another in a specific area. Okay? Saan yon? In London in 1854, nagkaroon ng spread ng cholera. Okay? Pag sinabi nating epidemic, ibig sabihin kung makalat siya sa napakaraming tao sa isang lugar. Okay, halimbawa sa London. Kapag kumalat na siya sa napakaraming tao sa buong mundo, ang tawag na natin doon ay pandemic. Si John Snow, there were large epidemics of cholera in Europe during his time and America that killed thousands of people. So maraming namatay because of cholera. Pag nagkaroon ng cholera, usually it causes severe diarrhea and dehydration. John Snow was a physician in London, so isa rin siyang doktor, who spent several decades studying cholera in a systematic way. So inaaral niya na yung cholera during that time. He is most often credited with solving an outbreak of cholera that occurred in London in 1854 and proposed a new hypothesis for how cholera was transmitted. He tested his hypothesis systematically by making comparisons between groups of people. He provided evidence for an association between drinking from the Broad Saint well and getting cholera. So napatunayan niya na yung cholera ay dahil doon sa kanilang drinking well. So ang ginawa niya, nagpropose siya ng intervention to prevent yung additional cases, and then dahil doon nagpropose siya na gumawa ng mga ibang sewerage system na dadaanan ng tubig na malinis. Okay? During that time, maraming nagkasakit ng cholera. Inaral niya kung bakit nagkakasakit ng cholera tong mga taong to, and then he proposed na dahil yun dun sa well na kung saan sila kumukuha ng tubig. Okay, so ang ginawa niya, nag-propose siya ng isang sewerage system kung saan dun dadalo yung malinis na tubig na hindi infected ng cholera. And then dahil doon, nabawasan yung mga sakit, and he was termed as the father of epidemiology or epidemics. Si John Tindal in 1820 to 1893, he discovered that there are two types of bacteria. So ito naman, unti-unti inaaral na nila yung different types ng microorganisms. Meron daw dalawang uri ng bacteria, the heat-sensitive organisms and the heat-resistant organisms. Ano yung heat-sensitive organisms? These are organisms that are easily destroyed by boiling. So pag pinakuluan mo yung isang solution, namamatay agad yung bacteria. Pero merong mga bacteria na nagkukos ng resistance against heat. Meron silang tinatawag na endospores. Tindal demonstrated that alternative process of heating and cooling, if repeated 5 times, can kill all the endospores. And ito yung tinatawag na sterilization. Ang sabi niya, para mapatay natin yung mga endospores na yan na dahil sa endospores, kaya hindi natin basta napapatay yung mga microorganisms or yung bacteria, dapat meron tayong alternate process ng heating and cooling, and then 5 times mo siyang gagawin. Ito ngayon ang tinatawag natin as sterilization or tinatawag ding Tindalization because he was the one who demonstrated this process. Okay, para ito sa mga heat-resistant bacteria na merong mga endospores. In 1876, a German botanist, Ferdinand Kahn, also discovered heat-resistant forms of bacteria. These bacteria are now termed endospores, Bacillus species, and Clostridium species. And then ano yung endospores? They are highly resistant, specialized structures that can withstand heat, dehydration, freezing, toxic chemicals, enzymes, and ultraviolet radiation. Pag meron ito yung bacteria, mas mahirap silang patayin dahil kaya nilang mag-grow on places na may matinding heat or walang tubig or sobrang lamig or yung mga chemicals and enzymes na naandon, kaya nilang i-withstand. Okay, so ang sabi nga kanina ni Tindal, para mapatay itong mga microorganism na to, dapat i-sterilize siya. So this one is an example of an anthrax bacillus which has endospores. Ano yung endospores? Yung mga bilog-bilog sa gilid niyan. Yan yung tumutulong para hindi agad sila mamatay ng heating, extreme heating. And then Paul Erlich in 1890s proposed a theory of immunity in which antibodies were responsible for immunity. In addition, he is known as the father of modern chemotherapy. Okay, he speculated about some magic bullet that would selectively find and destroy pathogens, but not harm the host. And he also developed a staining procedure to identify tubercle bacilli, which is the cause of tuberculosis. Okay, si Paul Erlich ay tinawag na father ng modern chemotherapy dahil sa na-discover niya as the magic bullet. Okay, ito yung drugs or chemical na ang itatarget niya lang talaga is yung specific cell na merong sakit. So kaya siya tinawag na magic bullet kasi meron lang siyang tinatarget. Okay, na specific cell kung saan na andon yung pathogen. In the process, napapatay niya lang yung microorganism pero hindi niya na-harm yung host. So yun yung tinatawag na selective toxicity or yung chemotherapy through the magic bullet. So ito si Paul Erlich, siya yung nagsimula ng chemotherapy. And this one is Alexander Fleming. He observed that the growth of the bacterium Staphylococcus aureus was inhibited in the area surrounding the colony of a mold that had contaminated a Petri plate. The mold was identified as Penicillium notatum and its active compound was named as penicillin. Okay, so si Alexander Fleming naman ang nakadiscover ng unang-unang antibiotic. Okay, meron siya sa laboratory na Petri dish na merong bacteria na tinatawag na Staphylococcus aureus. Ang Staphylococcus aureus is also reason ng pagtubo ng mga pimples sa balat ng mga tao. Okay, the same bacteria causes pimples. So yung bacteria na yon, ginogrow niya sa lab. And then hindi niya sinasadya na natapon yung mold ng Penicillium notatum doon sa bacteria na Staphylococcus aureus. And then he discovered na doon sa part ng bacteria or ng Petri dish kung saan natapon yung penicillium ay namatay yung bacteria na naandon. And this started the production of antibiotics, penicillin. Other scientists include Walter Hayes. He used agar hesse or his. He used agar as a solidifying agent to harden the media. And agar is extracted from seaweeds or red algae. So si Walter Hayes naman, siya yung gumamit ngayon nung ginagamit sa laboratory na agar. Okay, yung nilalagay sa Petri dish kung saan kinukuha naman yon from seaweeds or from algae. Yung pinakalalagyan ng agar, ang gumawa naman nun is Richard Petri. So siya naman, sa kanya naman pinangalan yung Petri dish. He made the agar dish to provide a large area to grow microorganisms. And then Christian Graham, he used a staining method that demonstrate bacteria and distinguish between gram-positive and gram-negative bacteria. Meron dalawang uri ng bacteria, gram-positive at saka gram-negative. To distinguish yung pagkakaiba nilang dalawa, ang naka-imbento ng process na yon ay si Hans Christian Graham. And then, dahil doon, mas madaling i-identify yung bacteria kung ano yung possible na ibigay sa kanilang antibiotics to kill this bacteria. So, the process, the one who invented the process to determine kung gram-positive ba o gram-negative ay si Hans Christian Graham o si Christian Graham. Okay, mapapansin nyo, pinapangalan sa kanila yung mga process o kaya yung mga bagay na na-discover nila in honor of this scientist. After that, Resca is the first electron microscope that was discovered. And this electron microscope is capable of magnifying biological specimens up to 1 million times. This computer-enhanced images of smallpox, number 1, yung una-una, yung kulay pink sa gilid. Herpes simplex and mumps are magnified. So herpes simplex yung nasa baba ay nasa taas yung kulay yellow, and mumps virus is yung nandun sa pinakababa. So this time, because of the invention of the first electron microscope, they were able to see how these viruses look like. They are respectively magnified 150,000 times and 90,000 times to study the virus structures in detail. So thanks to the invention of the first electron microscope, dahil doon, naaral na nila kung ano talagang itsura ng mga viruses. And then this one is Watson and Crick. They determined the structure of the DNA. And they used their research together with the research of Franklin and Wilkins to determine the structure of the DNA model. So the DNA model that we use today is because of Watson and Crick. So makikita nyo ngayon medyo mas in detail na, mas maliliit na details na yung nadidiscover dahil din sa pagkakadiscover ng electron microscope. So nakita na nila ano yung nandun sa loob ng mga microorganisms na yan. In the 20th century, eto naman si George Biddle and Edward Tatum. They studied the relationship of genes between enzymes using the bread mold. And then, eto na, yung bread mold is a fungi. So this time, fungi naman yung unti-unting nadidiscover. And Salvador Luria and Max Delbruck demonstrated spontaneous gene mutations in bacteria. After naman nito, nila George Biddle, unti-unti na-discover naman na nagkakaroon pala ng mutation ang bacteria. And actually, even by viruses, they mutate. Ano yung mutation? Nagbabago yung genes nila o yung DNA nila. Oswald Avery, Colin MacLeod, and Maclean McCarthy in 1944. Following the initial studies by Frederick Griffith, they provided evidence that deoxyribonucleic acid was the genetic material and carried genetic information during transformation. They work with Streptococcus pneumoniae, the rough and smooth. And then they discover na yung DNA ang reason sa bakit nagkakaroon ng genetic transformation or mutation. Okay, bakit nagkakaroon ng pagkakaiba? It's because of the difference nung kanyang DNA. Okay, so ganun ka-powerful yung DNA na kaya niyang magkaroon ng mutation or transformation. Ang pinag-aralan nila dyan ay ang bacteria na Streptococcus pneumoniae which causes pneumonia. In the 1970s, new discoveries in microbiology led to the development of recombinant DNA technology and genetic engineering. So dahil dun sa mga bagong advances na, may marami ng nadidiscover about the DNA, nagkaroon na ng science ng recombinant DNA technology and then yung science ng genetic engineering kung saan ine-explore na nila ano yung pwede nilang gawin sa DNA ng bacteria and viruses. Okay, these are the different scientists who contributed in the study of microbiology as a science, starting with Leeuwenhoek, hanggang kay Louis Pasteur, hanggang kay Robert Koch and the Koch's postulates, and then Joseph Lister. This is Edward Jenner. And then the next scientist, John Snow, Tindal, Ferdinand Kahn, and the next scientists such as Alexander Fleming and Watson and Crick. All these persons contributed in the development of microbiology as a science. And ngayon, meron ng bagong technology kung saan nagkakaroon na ng genetic engineering using these microorganisms. And now, nagde-develop na rin ng mga vaccines against dun sa mga bagong type ng viruses. So for your performance task, I will upload this on the Google Classroom later today. You have to construct a timeline of the contributions of the different scientists in the field of microbiology. I will give you an example. Halimbawa, ito. Pwede kayong gumawa ng ganitong example. May picture and then may konting descriptions and then meron siyang mga year kung kailan siya nangyari. Pwede rin naman na ganito yung itsura. Okay, hindi lahat lalagyan mo ng picture, yung mga very important lang. Pwede rin naman ganito yung itsura, yung picture lang ng scientist, yung ilalagay nyo. Or ano yung mga magandang bagay na pwede mong associate doon sa events nung mga tao na yun who contributed in the history of microbiology. Okay, please take note of the years kung kailan siya nangyari. Okay, you can use Google as a reference. You can use the PowerPoint presentations that I will be sending you and the ones that I have sent you earlier as your resource. And then I will send also the rubric. Ilan po yung minimum? Kung ilan yung nakalagay doon sa PowerPoint presentation ko. Anyway, I will be sending you the list na kailangan yung ilagay. Okay, so hindi nyo siya kailangan hulaan. Ilalagay ko yung list ng mga taong kailangan yung ilagay. And then, bahala na kayong mag-isip kung paano nyo siya gagawin. Ang deadline is, kailan nyo siya matatapos? Sige. When do you think can you finish this? Para masabi nyo naman na binigyan ko kayo ng ample time to finish the performance task. Two weeks, is that enough? Francis? And Rose, yes? Is 2 weeks enough for you to finish this?
Yes, Miss.
Okay, so that will be the end of October. That will be October 31. It's okay with you?
Yes po.
Okay, deadline of performance
Yes,
task number 1 is October 31. Baka naman mamaya kung kailan October 30, tsaka nyo siya gagawin, ha? Please manage your time. Binigay ko ng maaga, so gawin nyo ng maaga, okay? Later today, I will upload the things that you have to include and also the rubrics of your performance task. Okay? On Saturday, you can start with this performance task. So I will not be giving you any module for this week. So on Saturday, which is our asynchronous day, pwede nyo siyang gawin. So mag-attendance lang tayo ng Saturday and then gawin nyo na siya, umpisahan nyo na siya, mag-isip na kayo kung paano siya gagawin para hindi kayo mag-hold dun sa gagawin nyo. And then next week, we will start with the discussion of the types and the different parts of bacteria. Questions? This is individual, ha? Questions? Wala na ba kayong question? Okay, so no module for this week. You can start with the performance task. The due date is in October 31. And that's all for today. Good luck and God bless.
Thank you po.
Miss, thank you, Miss. Po.
Wait
Thank
lang,
you
may
po.
nagsasalita sa tulad.
Miss JPEG po.
Thank you.
Yung mismo, instructions pa pala sa Google Service.
Oo, isi-send ko yung instruction later today.
Thank you, Miss. Have a nice day.
Thank you, Miss. Have a nice day po.
Goodbye.
Thank you, Miss. Bye-bye po. Thank you, Miss.
Thank you, Miss.
Thank you po.